a revision video for required practical 6, and again reminder, the reason we're making these videos is because they are up to over 15% of the final grade will be based on these, so you do need to be prepared to answer detailed questions on required practicals for all biology students, regardless of whether you're combined or separate GCSE. This practical looks at required practical 6, which is photosynthesis and the rate of photosynthesis. Press pause, have a go at these questions, and then I'll go over them in a second. These are typical GCSE style questions, maybe more foundation tier than higher tier or lower higher tier questions. So you might more likely be asking a higher tier question to do the symbol equation, but let's have a look at it anyway. So carbon dioxide plus water and oxygen. Okay, so there's your answer to that one. Next one, light is needed for photosynthesis. The green pigment that absorbs light is chlorophyll. Please be aware the reason light is absorbed is it provides energy for photosynthesis. So because photosynthesis is an endothermic reaction which absorbs energy. And then plants need carbon dioxide and water and energy for photosynthesis. Complete the sentences. Carbon dioxide is obtained from the air or the atmosphere. Roots obtain water from the soil or the ground. Do not accept rain because they don't absorb it from the rain directly. It has to go into the ground and absorbed by the roots. And energy comes direct from sunlight. Then at photosynthesis and the basics of it. So you should know the word equation. If you don't know that, you need to think about what plants do. You should be aware just from your knowledge of the world, we've got an issue with too much carbon dioxide and plants absorb this. You should also be aware plants give us oxygen and they provide glucose for the which starts our food chains. Light is absorbed in photosynthesis by the chemical chlorophyll. So the use of chlorophyll is it absorbs light, which provides energy for photosynthesis. So the 3 limiting factors, there is a 4th one, but the 3 I want you to consider is carbon dioxide concentration, light intensity, and temperature. And they can limit the rate of photosynthesis because if there's no carbon dioxide, you don't have enough reactants. If there's no light, you don't have enough energy. And temperature affects enzymes, and enzymes will work slow if they're cold because they're moving slowly, or they denature at high temperatures because the active site changes shape. You should also know the symbol equation if you're doing higher tier, which hopefully you know by now, but it's 6 carbon dioxide, 6 CO2 plus 6 H2O goes to C6H12O6, which is glucose, plus 6 O2. You must know this symbol. Okay, so you must know glucose for your exam and be able to recall. So if we look at these limiting factors, the limiting factor is anything that can limit the rate of photosynthesis. And the 3 limiting factors are carbon dioxide concentration, light intensity, and temperature. So if we look at carbon dioxide concentration and light intensity, they have the same graph. So what this shows me is until about this point here, as I increase my light intensity, the rate of photosynthesis increases. But after this point, the rate of photosynthesis stays the same. So at point B on this graph, the rate of photosynthesis stays the same. So what you could be asked then is, well, what is the limiting factor at point B? Because it's no longer actually light. So the limiting factor can be seen like this. Whatever is the limiting factor here on the graph is a limiting factor for this bit. So as the graph goes up, it's a limiting factor. But if light is no longer the limiting factor from point X, at point B, the limiting factor must be either CO2 concentration or temperature. On this graph, carbon dioxide is the limiting factor until that point. So that is the limiting factor there. So the limiting factor after X must be light intensity or temperature. So it's the other 2. So if they say what else is a limiting factor, that's why you need to know all 3 of these, because you might need to mention one of the other 2 or both of them. The temperature graph is a bit different. This graph should remind you about enzymes, okay, and the fact that here it's a colder temperature. So here the enzymes are colliding at a much slower rate, so therefore they make less enzyme substrate complexes. This is the optimal temperature, and then here the enzymes get denatured because the active site changes shape and is no longer complementary to the substrate. You need to know some key questions for photosynthesis, so you should be able to answer these key questions about photosynthesis. So please press. Let's have a look at the actual answers. So photosynthesis is basically a reaction that uses light energy to make glucose. It combines carbon dioxide and water to make glucose and the waste product oxygen. It happens in the chloroplasts of cells, and mainly those cells are palisade mesocell cells in the leaves. It's a pigment which absorbs sunlight, is chlorophyll, which is that green pigment. The balance symbol equation is 6 carbon dioxide plus 6 H2O goes to 6 O2 plus C6H12O6 glucose. We can test the product. It's more chemistry of oxygen. If you collected the oxygen up, you could put a glowing splint into it and it would relight a glowing splint. And photosynthesis is important in a food chain because it makes glucose, which can be converted to other biomass in the plant, which then starts the food chain. Oxygen is obviously essential for all life on the planet because it is used for aerobic respiration, which happens to release energy from glucose. This is your basic method that you will be carrying out. So, and you should be able to recall this. This is a really common GCSE question. So you might be given a picture like this and asked what to do. So basically you would set up a test tube rack containing a boiling tube at a distance of, let's say, 10 centimeters away. Okay, you'd fill the boiling tube with sodium hydrogen carbonate. Now that makes sure that there's enough carbon dioxide in the solution. So it means that there isn't a limited amount of carbon dioxide. You would then put the pondweed into the boiling tube and then you would push it down to a glass rod. Leave it for 5 minutes to get used to the conditions, and then you would count the number of bubbles produced in 1 minute. Now this example here has actually come from an exam to show you how they might expect you to ask it. Now if you've got a question saying how could you limit the impact of temperature, there's 2 answers here. Use an LED light source, because LED has limited temperature release, or you could use a heat screen, so a plastic screen between the light that allows the light to come through, but not the heat. Collecting your results, you could be asked to draw a results graph. So if you're changing the distance because you're investigating light intensity, remember you need to put your centimeters in there as units. Never put units in the table other than in the heading, and then you would do a mean as you've run out of 3 averages. Okay. Independent variable in this investigation is your light intensity. Obviously that could be changed. You might be investigating carbon dioxide concentration, or you could be investigating temperature, but most common light intensity. The volume of oxygen produced per minute, and I would do oxygen rather than gas because sometimes they want to see it's oxygen being produced. And the control variables be temperature and carbon dioxide. So whichever of the 3 variables is not the independent variable, 3 limiting factors, is not the independent variable. The mass of pondweed could be kept the same. The duration of time recording oxygen production could be the same. The one I would avoid is the volume of water, and that is because of this. If you imagine you've got your test tube, there's your water. As long as the plant is under the water, it really doesn't matter how much water there is, whether there's lots more or not as much as a result.